Good morning, everybody. Welcome to the webinar. We're going to get started in a couple minutes as people trickle in. Okay, in the interest of time, I'm gonna do the introductions here. Good morning, everyone. My name is Ali Baim. I am a co-PI on this RCN. Some of my fellow PIs are featured here as panelists, and our two speakers are here, as well as postdoc Jill, who is working with us on the RCN. It's great to see everybody. We are starting off this academic year with the new series of webinars that are going to be monthly, and this is our first one. We're really happy to have Alessandro Zulli, who is a PhD student at Yale, and Smruthi Karthikeyan, who is a postdoc in the School of Medicine at UCSD. The topic of today's webinar is using sequencing to probe SARS-CoV-2 diversity in wastewater. And we're just going to jump right in here. But first, I want to explain the rules of the webinar. So we have a Q&A open, and please put your questions into the Q&A. As of right now, all of the attendees do not have the speaking function, although we can turn that on if needed. Questions should go into the Q&A, and you have the ability to upvote a question if you like it. And then we have moderators at the end of the 20-minute presentation who will ask the speakers the questions and moderate that. Okay, so let's get started. Alessandro is going to go first, and you can go ahead and share your screen and get started. Thank you.
All right. Good morning, everyone. I'm Alessandro Zulli from Pecha Lab, and I'll be presenting the results of a joint effort on predicting the abundance of SARS-CoV-2 from primary sludge samples. As a little bit of background, Yale and the Connecticut Department of Public Health implemented a widespread and timely system for the collection and analysis of wastewater samples early on in the pandemic. Daily samples are collected from 8 wastewater treatment plants spanning the entire state and serving a total population of just over 1 million people. This is a third of the population of Connecticut and covers all the major cities in the state. This program first started in New Haven and the surrounding area, but once this proof of concept was shown to be viable, it was quickly expanded to 7 other municipalities. The system has been used to help inform public health decisions in the state, and very importantly for this project, gives us access to a large array of samples spanning a large period of the pandemic, which is a key factor if we are to analyze the rise and fall of variants as the pandemic progressed. The Yale Connecticut wastewater program has shown that SARS-CoV-2 RNA concentrations in primary sludge track infection dynamics within the contributing population. In these first 6 graphs, we see the results of our modeling efforts based on this RNA signal. The colored line represent our estimates, while the gray lines represent cases by date of specimen collection. The equation in the center represents our final model, with the betas representing coefficients for the last 5 days of RNA measurements, and the gammas representing the known cyclical nature of daily testing. So using the RNA levels in wastewater, we are able to accurately predict cases within municipalities 5 to 7 days ahead of actual testing. We also maintain a website for the state which publishes SARS-CoV-2 RNA levels updated on a daily basis for the 7 municipalities we monitor. We also worked with Ed Kaplan, a professor of epidemiology, to develop a method for estimating the underlying infections within a population using RNA wastewater levels. These predictions represent the total amount of infections in our underlying populations, which is invaluable when tracking the spread of the pandemic. The predictions, as shown in blue in this second graph, are in line with estimations made by other accepted models of infection, which extrapolate from hospitalizations and deaths and other similar measures. So, the driving force behind wastewater-based epidemiology is the potential for efficiency gains that it can provide. Our previous work measuring RNA levels turned hundreds of daily COVID tests into a single sample summary of the local situation. In much the same way, wastewater-based sequencing can potentially reduce the need for vast and expensive sequencing efforts while losing little relevant public health information. Further, analysis of these wastewater samples can help us identify the geographic source of infections and the variants which are escaping vaccination efforts. To achieve these goals and to create an easily implementable pipeline, both laboratory and software techniques had to be optimized. Sequencing wastewater can be challenging due to several factors: the degradation of RNA in the samples, the high presence of inhibitory compounds, and the low concentrations of target RNA when compared to clinical samples. We had already developed a robust extraction methodology, so we focused on the sequencing methods. We worked to optimize the PCR protocols to our specific samples, focusing specifically on trying to minimize the sequences of RNA amplified while maximizing the replication to avoid some of the pitfalls mentioned above. To this end, we tried amplifying the entire genome as well as simply the spike protein, which, as you can see in the bottom image, contains much of the information necessary for variant determination. Now, on the bioinformatics side, the difficulties lie with the noise in our data and the misidentification of variants due to SNV calling. We implemented a software package called Callisto, which was originally used to quantify abundances from bulk and single-cell RNA-seq data to our wastewater-derived sequences. This program, unlike ones previously implemented to identify variants, does not use SNV alignment. It uses a method known as pseudo-alignment. SNV calling has several drawbacks, requiring high sequencing depth, creating a reference bias, and often ignoring the fact that SNVs occur in common combinations when you need to identify a variant. Pseudo-alignment therefore allows for far more efficient and specific identification of variants by identifying our sequences based on k-mers and assigning variants based on the intersection set of the resulting k-compatibility classes. So, let's go through what that last sentence actually means. Callisto's workflow is based on the idea of matching k-mers to reduce the computational complexity of assigning variants using Eulerian rather than Hamiltonian cycles. In simple terms, it turns a long string of base pairs into identifiable and overlapping chunks. It then uses this information to assign variants based on genome sequences without the assignment of specific mutations common in SNV calling software, such as vpipe. So the first part of this process is the creation of an appropriate set of benchmarks. Our reference set used nearly 1,500 sequences with 881 SARS-CoV-2 lineages represented. When running these benchmarks, we have multiple genomic sequences per lineage, which allows us to capture both in and out of lineage variation. As shown in the image, k-mers within a reference are used to create a set of nodes, which are then used to perform pseudo-alignment. In step 2, pseudo-alignment, the nodes are hashed to find the k-compatibility class of that specific read. This process importantly skips any redundant nodes to improve computational efficiency. A redundant node is simply a k-mer which is nearly always present alongside another particular k-mer and is therefore not specifically necessary for variant identification. The read is then assigned based on the intersection of these k-compatibility classes, giving us the final assignment of a single read. Then, in the final step, these abundances are filtered above a certain noise threshold, and they are summed up per lineage to report the final variant of concern abundances. For our work specifically, we created 33 benchmarks by simulating sequencing reads from variant genomes with varying levels of background noise. The true variant abundances in these generated genomes ranged from 0.05 to 100%, as shown in these graphs above. This was done for both the whole genome and for just the spike protein region, as that again contains most of the information necessary for variant identification. So in these top two graphs, we see the variant frequency within our reference set, the true variant frequency on the x-axis, and the estimates on our y-axis. One thing to note here is that we see underestimation across the VOC frequencies. This simulation also allowed us to determine a theoretical variant abundance threshold of 0.1% that would avoid any false positive. This is again only possible due to the lack of SNV calling, as sequencing within wastewater has huge variation in the abundance of specific mutations. Indeed, we should note that P1 is the variance most accurately identified in both simulations, which is due to the more unique polymorphisms that are associated with it. The bottom two graphs, shown here, show that what the relative prediction errors are at varying VOC frequencies. Now, we notice that at very low frequencies, the errors are relatively high, but at around a frequency of 1%, that is a true variant frequency of 1%, the errors stabilize. I would like to note that our method is particularly useful, for example, in the case of B1427 and B1429. These, if we were to use SNV calling or VPipe or other types of software, would be almost indistinguishable. But due to the inclusion of multiple sequences per lineage in our reference set, we can sort of get a big picture, holistic view and distinguish them. So for the sequencing, we adapted the Illumina COVID-seq diagnostic test using NebNext Arctic v3 primers. The PCR protocols themselves were optimized through standard trial and error to try and maximize replication by increasing the cycles or repeated PCR steps. The other approach we took to optimizing this protocol was to minimize the number of primers used while retaining as much identifying information as possible. The reasons for this are, again, the high presence of inhibitory compounds within our samples themselves. To this end, we again ran experiments sequencing only the spike region. So I would like to note, importantly, that CT values less than 31 yield at least 60% genome coverage with greater than 1 million reads. CT values less than 34 yield 40% genome coverage with around 500,000 reads. We wanted to see what the actual impact of this range of genome coverage would be. And to do that, we bootstrapped from a data set with maximum coverage, and we did it at random percentages ranging from 20 all the way up to 80%. On the right graph here, the results for B117 or the alpha variant are presented. And what they show is that while the median abundance remains relatively accurate, even at genome coverages as low as 20%, which would be a CT of around 35, 36, the uncertainty becomes large. Effectively, while the estimate itself may be accurate, it's relatively unreliable. Finally, we took the results of our analysis and compared it to clinical data. Clinical data, while imperfect, is our best estimation of the true abundance of these variants within a given population. In this graph, our estimates are in blue, while the green line represents clinical sample abundance. We see that the data for B117 represents an increase in the underlying population through April, while other VOCs, such as B1526 shown below, remained relatively stable throughout this time period. Both of these trends broadly match clinical data. Again, as we saw in our benchmarking experiments, we expected underestimation and expected the error in our estimations to be higher at lower abundances than higher abundances. This trend is observed in the resulting data, with wastewater predictions matching closely once abundances get to 40, 50, 60%, but trailing at lower abundances. I should note, this data used whole genome reads. While theoretically, spike-only sequencing could lead to more accurate results at low frequencies, In practice, we did not get enough reads. This was just due to incomplete genome coverage when using spike-only sequencing. We're hopeful that further advancements and refinements on the sequencing side could significantly reduce this problem and therefore increase the efficiency of wastewater variant abundance. So, to recap, wastewater sequencing has shown the potential for inexpensive, wide-scale application for the tracking of rising variants of concern, potentially reducing the need for expensive clinical sequencing or providing an option for areas that can't afford it. Our experiments have shown that abundances above 10% in the underlying population, as estimated from clinical sequencing, can be reliably quantified and tracked over time, particularly with improvements in sequencing efficiencies. On that note, we've shown that spike-only sequencing could yield better results with improved genome coverage. In practice, however, whole genome sequencing may still be necessary to capture enough information for accurate variant assignment. Lastly, we'd like to note the potential that these technologies have when adapted to both other variants of concern, such as Delta, and other viruses, such as norovirus and influenza, which can often mutate, and the tracking of their variants can be invaluable to epidemiology research. I'd like to thank all of our collaborators and highlight a few of them here. Dr. Bajans at Harvard was responsible for a lot of the applications of Callisto to our samples, and Isabel Ott at the Yale School of Public Health was invaluable in fine-tuning our sequencing process. I'd like to thank everyone for their time and open up the floor to questions. Thank you.
Thank you so much. That was an excellent explanation of your bioinformatic approach. As a reminder, please put whatever questions you have in the Q&A box. I'll kick it off. I guess I was curious in your simulation studies, or when you're looking at the variant detection, you had depth simulated. You simulated a certain read depth, and I was curious in your actual analysis when you're processing the samples, what kind of depth was required? Like how many samples after the PCR could you put on a lane, for example, in order to get the depth that you needed to do the variant calling?
So for these experiments, the results of our actual analysis showed that depths of around in the range of 20 to 100 times was fine for the actual variant calling and estimation.
Interesting.
And this is shown here on the y-axis if you were
curious.
curious.
Got it. So 20x. Yep. Okay. And then what is the turnaround time that you guys are doing for the samples, the sequencing and the bioinformatics?
Yeah, so we're lucky at Yale to have access to a great sequencing facility. Usually the library prep will take about 2 days. And then the sequencing itself, we will get the results about 2.5 to 3 days later. The application of Callisto itself is incredibly quick. It's maybe 15 minutes to process all of your samples, compared to something like VPipe, which took me 6 to 7 hours to run.
Cool. I'll take one from the Q&A box. Are you able to detect new variants with this approach? Seems like you.
So we are not able to detect new variants due to how the reference set is constructed. To actually be able to assign a K-compatibility class, that K-compatibility class has to exist within your reference set. Does that make sense?
Yeah. I have a question about when you were comparing to the clinical set. So it seems like you're kind of benchmarking your bioinformatics with the clinical data. Would you expect these in a perfect world of everything for these to align? Or do you expect there to be some biases in the clinical sequencing?
So that is an interesting question, and it's one we have thought about a lot. while we would expect the broad overall trends to match, we do expect that there is a bias in clinical data simply due to the fact that usually clinical data is based on more of the symptomatic cases and perhaps some of the more serious cases that actually get sequenced, since sequencing is a limited sort of resource.
Right. Okay, and then what population is covered by the sample collection? So is this centralized wastewater or is this neighborhood level wastewater?
So the towns we cover range anywhere in population from about 18,000 to 200,000. Norwich, New London are smaller towns where a family getting COVID can easily show up in the wastewater, while, for example, in Hartford, it's sort of harder to move the needle. But these are centralized. All of these are from wastewater treatment plants because we are using primary sludge for this analysis.
Okay. And then, okay, does the bioinformatic approach have the potential to conflate variants, or does it require a unique SNP?
I'm sorry, could you repeat the first part of that?
So this is from Kyle. So does it have the potential to conflate variants, or does it require a unique single nucleotide polymorphism?
Oh, so it does have when you say conflate variants, I assume he means sort of putting together several different lineages. And we do that in the third step of Callisto right here. So each reference creates its own sort of reference set, its own K-MERS and K-compatibility class. And then once we've assigned it, we filter them based on a noise level that was determined theoretically as, I think, 0.01%. And anything above that gets summed up under the sort of heading of a per lineage basis. So B117, for example, might have 18 different genomes in the reference set, and all of the reads that are assigned to those 18 different genomes then get summed up to find an abundance per lineage.
Okay. All right. This is a question about the model comparison when you're comparing your wastewater monitor to the machine learning and the hospitalization model. So one question is, how does the wastewater compare with respect to error or uncertainty? So while the estimates all seem to line up across these models, one question is the error bars with the wastewater model larger than a hospitalization model, for example?
So that's a good question. I don't have the error bars for these other models off the top of my head, but from the last time that I have looked at them, they are also fairly wide. I would expect wastewater to be a slightly wider confidence interval just due to the noise present in the samples.
Okay, we're just like zooming through these, so feel free to put your questions in the box. This is a question from Rose, which is, given how this is going, how often do you have to update your reference set?
Yes.
How are you going about that?
So we try to update our reference set fairly often. We're very lucky to be working with Dr. Grubow at the Yale School of Public Health. They do pretty much all of the sequencing for the state of Connecticut, and so they can provide us with up-to-date data pretty much as soon as they have it. I think Yasmin last updated our reference set a couple days ago. As far as how often you should update it, that will still kind of depend on the results of clinical data. You'll still need to do clinical sequencing to figure out what these variants that are sort of showing up are, and then apply it to this sort of modeling.
Awesome. Okay. Let's do one last question, just in the interest of time.
Sure.
Which is one comment, I guess, is that Delta is probably going to become the common ancestor for all future variants.
Yep.
And so the question is basically, how might that affect this approach? If you're just updating your reference data, maybe it doesn't matter, but does this take into account when you're thinking about these new lineages in the common ancestor? Is that going to affect your approach?
So if it becomes of interest that Delta becomes the sort of common ancestor, there will still be variations within Delta itself. And because of the way that our approach is structured, where we use a reference set with a lot of sequences per lineage, we would actually be able to distinguish between specific references of the Delta variant within the Delta lineage. So you would simply construct a reference set for whatever the progeny of the Delta variant are, then be able to assign it. And if you were interested, you could effectively grab the data in the second column here, where it's abundance per reference, and be able to distinguish, oh, this Delta variant offshoot is much more virulent or has a higher infectivity or something like that.
Awesome. Well, thank you very much for sharing your approach. I know there was a couple questions that came in the Q&A box after I said it was the last question. So feel free to address those questions in the chat, or I'm sure I'm going to go ahead and volunteer you. People can contact you via email, I'm sure. But thank you so much for the excellent presentation.
Of course. Thank you for your time.
Thank you for that great talk. Okay, our next speaker is Smruthi. You can go ahead and share your screen. Thank you.
Are y'all able to see my screen?
Yes. Yes.
Perfect. You can, I'm guessing you can hear me as well. Hello, everyone. This is my first time at the seminar, so it's really nice to meet all of you. I am a postdoc in Rob Knight's lab at UC San Diego, and before I jump into our sequencing data directly, I want to give you a snapshot into where our sequencing data actually comes from. So here we are, switch testing is threefold. We do switch testing on UCSD campus. We also do it for the San Diego County school districts, and we also do it for their main wastewater treatment plant. So for the school, we had a program called Return to Learn, and we started this program in November of 2020, and it's still going strong. We've processed 21,000 plus wastewater samples from our campus. And for the SASE school districts, the program is called SASE, and this one was conducted in tandem with surface sampling as well as diagnostic testing, which was offered free of cost to all the students and staff in the school. And lastly, we have the San Diego County. So unfortunately, San Diego County is a large county. So the catchment area is about 2.3 million residents. And as you can see, that is just really large for us to get into high-grained or high granularity into our sampling analysis. That is why we use UCSD campus as a mini test bed for San Diego County. So why did we start in UCSD campus? It was mainly because we had a lot of students living on campus despite the pandemic, and UCSD decided to keep all of them on campus. So that's when we started a Return to Learn program, where wastewater was the main component driving the administration to send out notifications for getting tested. So the way we organized our campus wastewater system was we have about 130 auto samplers, which collect time-weighted composite, and they cover 360 residents and non-residents buildings on campus. This gives us 100% coverage of all the buildings with occupancies on campus. Out of it, 72 are residential, 4 of them are isolation dorms, which we use as a positive control and for other research-based questions. And 52 are non-residential. As of last week, our campus is fully open, and we have about 65,000 on campus on a daily basis, because our medical campus is also in the La Jolla campus, and 18,000 students are currently living in campus housing. So this is basically a map of UC San Diego, and all the buildings you see are UCSD buildings, which are denoted in gray, and all of those tiny orange circles are where we have our auto samplers. As you can see, we pretty much wanted to cover the entirety of campus using our wastewater surveillance. And this just is a snapshot showing the coverage in residential areas. Zooming in on one of these, you will see this is our most populated graduate and family housing. And the way we designed manhole to sampler ratio was based on occupancy data and agent-based modeling. So for instance, one of these buildings, AS069, has a building per sampler because that building alone had over 500 residents in there. However, if you see at the bottom, you'll have one for many buildings. That's because we wanted to have at least 150 people per catchment area for a sampler, so it doesn't get too crazy. And the way we split it up is in routes. So we cover like 7 routes a day. So we get all the samples on a daily basis. And the way we want to make sure that our samples and our samplers and our buildings are linked are by unique barcodes. We slap one on the auto sampler ID, and these are pre-linked to the buildings that drain into them. So this way, we don't have to keep worrying about if we freaked out the wrong building. And every time our volunteer or our staff go to pick up samples, they just pull out a mobile app, and then they scan the barcode and the sampler and the sample bottle. So it already gets linked back to the buildings which would drain into it. So we currently use a high-throughput SARS-CoV-2 viral detection pipeline. We don't do any pre-filtering of any kind because our samples are super upstream and we don't want to lose the solids. So the reason we had to do a high throughput approach is because the admin and basically UCSD relies on our wastewater data to make campus decisions. And it is imperative that they get all 130 wastewater samples analyzed, and they want the data on the same day. So for us, all of the samples along with the county samples are all processed on the same day. We have a 5-hour turnaround time from when we get our samples to when we get our results. And most of it is hands-free, so it doesn't take too many people to run it. And it's pretty much, we don't see, it's pretty much reproducible because we don't have too many human errors going along with this. And once we get the data, it's pre-linked. We already did the back end where we linked the building occupancy data, which for residences do not change, but they do for the non-residence buildings. So we pre-link these to our unique sampler IDs. So every time we get our qPCR data, what you see on the left is how we have our data in our Google Sheet. And what you see on the right is the public-facing view, which is our UCSD dashboard, which gets updated every day. So the left just shows each sampler's unique ID and the CQ values for that day and the status of the sampler. The backend programming just converts what you see on the left to the right. So this is what most of them use before coming to campus. And now we're trying to actually see if we can go into more detail for people who live on campus because we have an increased occupancy, and we're trying to see if we can get into more granularity, and we're currently testing this out. So the way our entire system works is we automated the notification and testing, and every time we already have the building occupancy and the roster. So every time the wastewater triggers a positive, targeted notifications are just sent to that building. And when we started this pilot program, every single person on campus was mandated to participate in asymptomatic nasal swab testing to validate our wastewater data. Mainly, we wanted to see if we missed anything in the wastewater that was captured by the clinical testing. In spite of that, we found we detected 85% of campus cases early just by wastewater, in spite of having regular diagnostic testing. So with this, I want to make a segue into our sequencing efforts. So this plot just shows how our wastewater data correlate with our diagnostic data. and we get an extremely high level of correlation between our wastewater as well as our campus nasal swab data. So the line you see here, anything left of that was our pilot phase, where all the students as well as staff who were on campus had to be, were mandated to test once a week. And after which, once we did the validation and we found that we didn't really miss anything in the wastewater that was captured by the diagnostic testing, we moved to response testing phase, where you would only have to test if you got a wastewater notification from us. And we found that within 1 or 2 days, we were always, almost always able to identify the case associated with that building, which gave us a wastewater signal. So this gave us the impetus to start tracking variants, mainly because UCSD, at least for the purpose of wastewater testing, can be considered a closed system per se, because our Point Loma is too big a catchment for us to make any actual decisions. So we decided to sample all our campus and county wastewater. Our county wastewater consists of the school district programs, as well as the Point Loma wastewater treatment plant, which the county wants us to keep sampling to get a higher level perspective. So for us, we found CQ does not really matter. We get near 100% coverage, genome coverage with pretty much all our samples. Our minimum, I think our lowest coverage was 75%. And we think that it's because of our upstream processes on how we concentrate and extract our RNA that gives us a higher RNA quality. But again, this is just a guess, so I'm not sure why exactly, because we did try some other ways of concentration, which gave us heavily fragmented RNA, and we could not get enough sequences. That's why we switched to the nanotrap in the hopes of getting more longer fragments, or at least closer to intact RNA. So the CQ values for all our, I've only shown a subset of 500 samples. We have about 2,200 to 2,500 sequence. I took a random subset, and the CQ values are shown in blue. Our average CQ value for wastewater was 34.6. Unfortunately, as you can see from here, very few of our samples were below the 30 cutoff, which initially the sequencing course said they needed to be able to sequence. But that would mean that we only had like 3 or 5% of our samples left. So we used a different protocol. We sequenced pretty much all the positives we got, irrespective of CQ. And our median genome coverage is 96%, and mean is 93%. And our reads are capped at 2 million. So we run 1,532 wastewater samples in one NovaSeq run. And the wastewater samples have its own run. Their libraries are prepared separately. They do not go with our clinical samples, just to make sure we don't have lane contamination. So in order to also make sure we don't have lane contamination, we add several controls to our sequencing plate to make sure that the ones that are designated empty or negative do not have any SARS-CoV-2 reads, which would bias our data. So from this, we collected all of campus and county wastewater data, which you see on the left. We collected from a period of November, and we're still collecting. So you can see about 9+ months of data on the left. So the left is all our wastewater data, and the right is our county nasal swab data. So we're also fortunate enough to be processing all the county samples, nasal swabs, as well as campus sample nasal swabs. So we are able to have a rich data set to compare directly. And the right is about 16,000 samples from nasal swabs from San Diego County, which we processed here. And we work with Christian Anderson's lab at Scripps, and we also get the samples from them to compare. And they have been helping us with the sequence analysis as well. So on the left, what you can see here is you can see the early onset of Delta in the wastewater. You do see a sliver of it in the county samples, but it doesn't come up till later. For us, we see the prevalence increase steadily before it completely took over, and that is mirrored in the county nasal swab data as well. So moving on to the campus wastewater data, this is just our wastewater sequences from our campus. So here you can see a really nice transition from when we started in November to how we are right now. And you can also see mu right on the top. So we hadn't detected mu in any of our nasal swabs yet, but we picked it up in wastewater. However, it was flagged as a regular lineage as 1.6 point something. We did not know that was a VOC until the WHO had classified it as one recently. So while we did have mu, we just didn't know that was mu till later. So we just flagged it as other. So this provides a strong case for also tracking emerging variants and using genome sequencing for doing that. And this shows our campus nasal swabs compared to our wastewater. So every single campus positive was sequenced as long as their CQ values were below 30 for nasal swabs. For campus wastewater, every single, all of them were sequenced. We kept the sequence number low to remove duplicates. So we did have a lot of replicates, especially if it's isolation dorm, because a person from one of the buildings gets moved to an isolation dorm. So we found that this provided us with really good concordance, but it shows the appearance of all of these signals earlier in wastewater. So if you see the pink there, which is Delta, and we see the pink coming up way before we actually found it in the nasal swabs. So this gave us a little more confidence in using this type of genome sequencing to say, okay, maybe, even if it's an emerging variant, or even if a variant is at very low proportions in wastewater, we can still pick it up, and we don't need a minimum amount for it to be able to be picked up. And this is just taking each variant by itself and is showing how our nasal swabs compared to our wastewater swabs. We found a very good correlation between our nasal swabs and our wastewater swabs for campus. And for a lot of times, like you see with Alpha and Delta, we found it earlier in our wastewater data as well. And the right just shows the frequency of samples and how many were assigned to each lineage. So now we get to a little more of an interesting part. So since we had all the wastewater genome sequences as well as the campus nasal swab genome sequences, we try to look at the phylogeny, and we do find identical matches. And we do know that it was the same person in the building who contributed to the wastewater signal. So if you see a blue, it means it's a wastewater sample. If it's a gray, it means it is a nasal swab from campus. And this is just zoomed into the same one for just showing just Alpha, because I'm unable to show the whole thing here because the phylogeny is too big and it's too busy. And, I'm sorry. And the same for SARS-C samples. So this also helps us see if there were multiple people shedding in one building contributing to a wastewater signal, or if it's the same person who's been shedding long after they did not test positive for their in their nasal swab. This is mainly a case here that you see on the left. It was the same person. They had been identified, but they were still coming back to school. It's just that they were shedding it in their stool for a longer period of time than average. And this especially is useful for SARS-C because our consent rate varied, and this would help identify outbreaks to see if there are multiple people who are contributing to a wastewater signal, or if one person was, if one of them had a party and gave the same strain to others. So that's where we're at now. Finally, I just want to end with how we distinguish our variants in wastewater samples, which have multiple strains, which clinical swabs do not have. So their diversity is much lesser and it's straightforward. With wastewater, it is not as straightforward. So to do that, we did a lot of experiments where we actually mixed strains in the lab and then we sequenced them. We did not do in silico mixing, which means that we did not mix sequences and then use our algorithm. We actually mixed the strains because we know that there are a lot of artifacts in sequencing that would be needed to be accounted for. And we did, we mixed about 5 different strains in different proportions. And we found that we used a barcode algorithm. We found that every single one of the times it predicted exactly what we expected to predict. And as another line of evidence, we also used a VOC detection kit, which is RT-qPCR based, which specifically targets mutations. So similar to our synthetic mix, we also pooled our wastewater samples, which we knew were pure lineage because of just one person in the building. We mixed them in different proportions, and we ran our RT-qPCR, and we found that it had a high level of correlation with our sequence data. So this helps us to fine tune our previous algorithm that you see here to make sure that this is not only useful for ideal cases where you have mixing with clinical samples, but also more environmental matrices like wastewater, which we know are more complex and can have inhibitors and all sorts of junk that can impact the way we interpret the data. So with that, I would actually like to conclude my talk, but I really want to take a moment to thank every single person who made this possible. This was a big undertaking, and I don't think I could have done it without the help from any of these departments. And if you have any questions, I would be happy to take them now. I hope we're good on time.
Yes, we are perfect. Thanks so much, Smruthi. That was a great talk. As a reminder, if you have any questions, you can add those into the Q&A box. And I'm going to start with the questions that are there and work my way through. So the first question that we have is for your sequencing data with the different variants, how did you determine % of each lineage in your wastewater samples?
That's a great question. So that goes back to, I hope you can see this. So we use a barcoding approach and we solve for an equation AX minus B. So I am not a mathematical modeler. My background is on computational biology. So this was done by our collaborator in the Christian Andersen lab, who used a barcoding approach to solve for an equation bounded by 1. So this takes into account all the mutations associated with a single lineage, as well as all the lineages and prevalence are taken into the training data set, with some leeway for other mutations that could happen for emerging data. And they are solved for by using the equation below. I can definitely get more data on this, and I'll be happy to send you this data, but we're still in the testing phase, and we just sort of fine-tune this algorithm last week. And then based on the mutation, we use something sort of, since we have near complete genome coverage for most of the samples, we don't believe we're missing most of the variants that we think would be at low abundances, which is why we made synthetic mixes ranging anywhere from as low as 2% to 98%. So we started at 2% and then ramped it up all the way to 100% to see if there is a mix in wastewater that has an abundance of, let's say, only 2%, it would be likely engulfed by the 98% unless you have near complete coverage. So to make sure that the algorithm can still tease apart that is why we did these synthetic mixtures with 2%, 3%, 5%, 15, 20, et cetera, to see if it's still picking up in all of these proportions, because you can see here, we went all the way from I want to say 2 to 100. And that is also reflected in our plots here. So we have to remember one thing with wastewater, that if a person is still continuously shedding, that will still be picked up in the wastewater, especially if you take something like point loma. So here, when you see the alpha that's completely gone in the nasal swabs, you will still see it trickle down here. It's just because it's the same person who's still on campus shedding. So this is how we used our algorithm. And then we directly compared this to not only a clinical swab data, but also all of these samples were run through our VOC panel to see if there's some sort of concordance. Unfortunately, our RT-PCR VOC panel, we can't use as quantitatively as we like. We use it semi-quantitatively. But yeah, it's mostly by looking at multiple different lines of data. But our main test bed was this synthetic mixing and then sequencing. We're hopefully going to write up the convolution much easier and then put that on our GitHub. All our sequences are on GISAID already. They're public. A couple hundred are already out on just say some of them are still in line. So feel free to download any of our sequences. All of our campus positives have also been deposited. And we also have our complete pipeline here. Our pipeline is also high throughput. So it takes a processing of, we process about 15,000 samples in a run. It only takes an hour to assign the pangolin lineage. So there are some times where our QC would fail because pangolin cannot assign a lineage because there's a mix. But in that case, we look at our actual sequence data. If that has passed QC, we then use it because pangolin expects only one and wastewater does not. So in that case, we just go by our actual sequence QC and not what pangolin expects. But we run both anyway, just to see. And pangolin requires a minimum 70% coverage.
Okay, thanks so much. Yeah, I think a lot of people are trying to, parse out these details from sequencing data, and it's great to hear people's perspectives on how to analyze such a messy sample type and messy piece of data. So the next question is a little bit more logistical on sort of how you're applying this data and relating it back to clinical tests. So did you look, or did you take into account any lag for clinical testing since the wastewater notification?
Our campus is 12 hours. Our campus turnaround time is 12 hours for all samples. We process 12,000 a day, so it's 12-hour turnaround. For
the clinical or the wastewater?
For, you mean sequencing? Oh, wastewater is 5-hour turnaround. Campus nasal swab is 12 hours, assuming they, tested that day.
Okay. Yeah, that definitely answers that question. Both of those are really quick turnaround.
County is different though. So county we can't comment on because we don't get the samples as quickly. Wastewater we process the same day, but our county nasal swabs by the time they reach our sequencer is not as as high a turnaround as, not as quick as we want it to be, but that's why we use our UCSD data to guide.
Sure, yeah, that makes sense. Okay, so the next one is back on looking at your lineage data. So it seems that wastewater is at times detecting lineages, not just earlier than clinical, but in cases where they're not present in clinical. Actually, you might have already addressed this in your answer to the other one. What's your hypothesis as to why some of these minor lineages are in wastewater and not always in the clinical data?
So a lot of times we do find it in the clinical data just later, because if it's residents, we find it much faster. A lot of times, most of the mu and lambda we detected early, it was because, we did not mandate testing them because everyone was vaccinated. We have a 95+% vaccination rate. And it's mandated on UC campuses as well. So a lot of times it was because a person might have been asymptomatic and did not test. And then after repeated wastewater notifications, they did test. In the beginning, we did not mandate. So we still don't mandate testing from a wastewater notification if it's non-residents. Only residents are mandated to test post wastewater. For non-residents, because it's hard to keep track of who comes in and goes out of the building, we don't. So that is more of a hit or a miss if we can find that person actually tested. If it's an employee, for example, our mu and lambdas were all from buildings which were more, campus facilities services or something like that, which were not residents or research buildings. So in that way, this actually, makes the point for wastewater that in spite of not detecting it in clinical samples, you are getting it in wastewater. So this actually, I would say, makes the case for wastewater, if you will.
Great. All right, so the next question is looking sort of over time at your data. Have you noticed any notable change in area under the curve for clinical versus wastewater between alpha or delta prevalent time periods? So as possible evidence of maybe different shedding characteristics between the different variants. I know that's something that people are discussing a lot these days.
Right. That's an excellent question. We're currently doing that for isolation dorms. So we have isolation, we're separating the isolation dorms where we know the exact number of people. And we are also comparing their clinical swab data. For instance, for Delta, we had a fully vaccinated person who tested positive for the Delta variant and their nasal swab CQ was 11. So we're trying to see if that's something we can use with wastewater. With CQ values, it's hard for us to say because we also sample stool samples from these asymptomatic individuals. And we don't find an exact, it's hard for us to say because you're asymptomatic, you will shed less. We didn't actually find that. We found that an asymptomatic person shed just as much in their stool. So right now, that's something we're focusing just on the isolation dorms and hope to have some data soon.
Great. So next question is sort of on your whole ramp up of the campus approach. I also noted how the barcoding approach seems like a really great one for keeping everything straight when you're ramping this up on short time frames. So curious if you've published that approach for sample collection or if it's available to try out anywhere. Is that also?
So we are currently, actually the barcoding approach is the one we tried out last week after trying a bunch of different approaches. And this was the one that gave us the most correlation. So this is very recent. That's why you'll see that slide is like hastily put together. I'm sorry. But yeah, hopefully we normally, before we publish, we make all our data public. So we would, we're planning to put it on a GitHub repo for others to try it out. And they are welcome to use our samples. I can send you the GISAID accessions for all our samples. It has our school samples, our Point Loma samples, and our campus samples. So hopefully once we put that in our repository, I'll definitely share it.
Great. And then also on the high throughput side, someone wanted you to comment quickly on what your concentration method is to
our
process that many samples.
It's that one we published recently with our campus manuscript, and it's also available in protocols.io. We put even the programming for the Kingfisher, everything in there. So it's reproducible as such.
Perfect, thank you.
PCR is constantly evolving though, so
okay.
keep adding VOCs and stuff, but yeah, our original methods are all in there.
Perfect, thank you. And so maybe we have time for one more question here. So, and this is, I think, touching on something that a couple of people have asked. So for the surveillance that you're doing, do you plan to continue offering these services to your university and schools indefinitely? And or are you planning to expand them to different locations or translate them into industry? Sort of what's your perspective on sort of the long-term longevity of your approach?
That is actually a really good question. So right now, San Diego County extended our contract. So the SASE program for schools has been expanded significantly. We just started our installs, and all for everyone who's consented, staff or students, a UCSD doctor and a nurse will go test them every time we get a wastewater signal the very next day. And this is completely free of cost. And the only thing is the SASE schools are chosen specifically based on their income level and the area where the vulnerability index are very high and they don't have access to proper testing, or they have a lot of social factors that prevent them from wanting to go get tested. So currently they're only offered in areas where, the prevalence rates are high, hesitancy to vaccines, and testing rates are also pretty high, and where most of them are very low income. So those are the areas currently covered, but we've expanded to a lot more schools. This academic year, our campus as well as our SASE program and the Point Loma will be going on at least for the academic year. But beyond that, we're hoping to maybe make this more modular, and we brought down our sample processing cost significantly. It's $13 all inclusive for a sample. So we're planning to offer this to more schools or places that want to do it. So we haven't exactly offered to industries at this moment because we are a research lab. So we can't process for companies or others. We can do it for academic reasons or for the county. And also since we do it for research, we can't charge anyone who we do the analysis for. So we offer it for free for the county as well as the schools. We haven't commercialized it. No.
Sure. No, just curious. I know that a lot of people are have these large programs up and running, and it's all uncertain. Everybody's sort of working in different limitations on how it can continue and where to apply it. So
instead of commercializing, we made it open source, so our whole thing just can be applied, and we are willing to help like universities and stuff set up, because that's more on an academic side.
Definitely. Well,
but next academic year, so that's
right.
Beyond that.
Yeah, it's always hard to predict. So I think we're right on time right now, so I'm going to close out our webinar. Thank you so much to our two presenters, Alessandro and Smruthi, for sharing your work with us. These were really great presentations, and I hope everyone enjoyed it. I've put a link in the chat if you want to click it before you leave. That's where we post our upcoming webinars and recordings of this webinar for anyone who will want to take a look at that later. And we should have that posted later today. So our next webinar will be in about a month, and it'll be on that site. And we hope to see you there. Thanks everyone for joining us, and have a good rest of your days.
Thanks for having me. Bye. It was nice meeting all of you.